Okay, guys, now we're going to talk about the organization of DNA. So the complete DNA sequence in every cell of an organism is called the organism's genome. Within the nucleus, DNA molecules are packed into these thread-like structures called chromosomes. The length of a DNA in a human cell is nearly 2 meters. But the DNA must fit inside of a very, very tiny space within the nucleus. It's only about 5 to 10 micrometers. So how is that possible? How can 2 meters of DNA fit into this small place, all right? DNA is basically tightly bound into small proteins called histones. These histones help package the DNA into chromosomes so that they can fit inside the cell nucleus. So look at all the DNA that can be found in this chromosome, all right? It's all packed tightly in there. Then you unwind the DNA, all right? And you unwind it again, and then you start to see, as you keep unwinding it, you'll start to see your double helix DNA, that ladder with the backbone phosphate sugar and the nitrogenous base pairs, all right? The guanine and cytosine and adenine and thymine base pairs. So these histones, these proteins, basically take the DNA double helix strand and they package it up nicely so that it fits in a chromosome, so it can fit into the nucleus. Although the number, size, and shape of chromosomes vary considerably between different species, their basic structure is the same in all eukaryotes, all right? So humans have 46 chromosomes, all right? Take a look at a chicken. They have 78 chromosomes. And a fruit fly, they can have 8 chromosomes. So each cell, and you have billions of cells in your body, each cell contains 46 chromosomes, all right? That's pretty cool. So each of these 46 chromosomes in a human contains thousands of genes on them. Remember I told you, here's your double helix strand, all right, that is tightly packed up in a chromosome, and it's all unraveled now. Each part of this double helix strand contains segments, and these segments are known as genes, all right? So each protein, okay, let me just back up. So each gene, okay, is a segment found on the DNA. It's located in specific places on the chromosome, and they provide the necessary instructions for creating proteins. So here we've got one gene found on the double helix DNA strand. This gene has specific codes, all right, specific nitrogenous base pairs that basically create instructions for creating specific proteins. So this gene, number 3, might create, with its genetic code, all right, a specific protein. But this gene over here might have a different genetic code, a different kind of arrangement of nitrogenous base pairs on its ladder to create a different protein. All right, this protein might carry out certain instructions, and this protein made by this gene number 3, carries out different instructions. All right? Okay? And this gene number 1, creates a different protein that will carry out different instructions again. All right? So each segment of gene creates a protein because of the code that is found on the gene to perform a certain kind of task, a certain function. Some of these tasks, all right, some of these things that the proteins do, well, one protein might help build up parts of the body. Another protein might carry out materials within your body, all right? Another protein might transfer signals from one body part to another. Some are called enzymes that speed up various chemicals in your body as well. So these genes, these segments found on the DNA, determine what kind of proteins your cells make, and those proteins carry out specific tasks. And therefore, these gene segments on your DNA that create codes, which create certain proteins, basically control everything about your body, how it functions, and how it looks. Pretty cool, eh? Our body's pretty amazing. All right, so now let's take a look at the human genome. The human genome, like I said before, contains 46 chromosomes. Those 46 chromosomes are arranged into 23 pairs. So therefore, there are 44 or 23 pairs of autosomes. So here, 1, 2, 3, 4, 5, 6, 7, 8. I'm just going to bear with me for a minute. These ones that I have circled, there's 44 of them in total. So if I go 1, 2, 3, 4, 5, 6, and I continue all the way down, I will get to 44 each individual chromosome. But there are 22 pairs that I'm talking about. All right, so 44 individual chromosomes and 22 pairs. Of the ones that I'm circled, the 22 pairs and the 44 individual chromosomes, these are called autosomes or non-sex chromosomes. All right? So the 22 pairs that I circled are non-sex chromosomes. Autosomes is the fancy way of saying that. The remaining one right here, it's called X. All right? There's 2 here, 1, 2, and they're both found under the on top of the X. These are the sex chromosomes. So this one here, these 2 chromosomes, this one pair is the sex chromosome. So you have 22 pairs of autosomes, or 44 individual chromosomes, and 2 sex chromosomes, or 1 pair of sex chromosomes. And these are important because this determines the sex of that organism, okay? So a female contains 2 X chromosomes. So here we go. This is your female. The sex chromosome, all right, if it has 2 X's, it's considered female. If there is an X and a Y, like right here, then that presence of the Y means that it's male. So this is called a karyotype, all right? That's a karyotype. It's just a way of looking at all the chromosomes and chromosome pairs, all right? So the diagram to the right is known as a karyotype. It is simply a picture of a person's chromosomes. So what sex is this karyotype? If it has 2 X's, can you please tell me if this karyotype belongs to a male or female? Well, if you said female, you would be correct. This is a female organism because it's got 2 of the X chromosomes. If it had the Y and X chromosomes for the sex chromosome, then it would be male. Okay. Please watch the video on DNA if you want. It's a YouTube video. You can look at that for more information. We're now going to move on into mutations. So the specific proteins that the genetic code made back on the DNA strand, double helix strand that I talked to you about, depend on the order of the DNA building blocks in that gene. So remember those nitrogenous base pairs? The order of these nitrogenous base pairs creates a gene, all right? If there's a change, a substitution, a deletion, or addition in that order of those nitrogenous base pairs, that is called a mutation. Okay, so let's take a look at the definition of mutation. A mutation is any change made to the DNA gene sequence. Okay, so if I go back here, all right, there are, where is it? Here. If there is a change, a substitution, deletion in that genetic sequence, which I told you right here. So there's the genetic sequence here. If it was like A, T, C, G, if something is changed in one of these, if it gets substituted out or something's added or something's deleted, then your protein that it makes gets changed, and therefore the function gets changed, or it doesn't work properly. That segment of the gene doesn't work properly. So going back here, okay, the mutation is a change made to that DNA sequence in a gene, all right? There are two kinds of mutations. Let's talk about the first one, a point mutation, okay? And point mutations can happen three ways. You have a base pair substitution, all right? So if this is my normal DNA sequence, all right? This is my, what is supposed to be the gene, all right? So it's supposed to be AGTCGA. Don't worry about codons. Those are just the codes that specifically make the protein, all right? A point mutation is I'm supposed to be AGTC. Uh-oh, it's not C anymore. It's become A. So if it's been substituted from C to A, now that's going to create a different protein. That's going to carry out different instructions, okay? Another kind of mutation that's a point mutation is insertion. Now, insertion, if it's supposed to be, once again, AGTCGA, it's not G, it's now T. And now there's the G, and then there's this T, and then there's the C, and then there's the G, and then there's the A. So this guy got inserted in, he got added. So now you have a different code. So you have one code, two codes, three codes, instead of just the one and two codes, which is found in the normal. All right, so I'm just going to erase a bit so we can see the last point mutation. So that was insertion. Now let's look at deletion. So once again, this is my normal DNA sequence. So if I look at deletion, I'm supposed to have a G. There's no G. It got deleted. So this is another kind of mutation. So point mutations can be substitutions, insertions of DNA base code, or a deletion of one of those letters. All right. What causes a mutation? Well, mutations can be random, mutations can be uncontrolled, and mutations can happen for absolutely no reason. Spontaneous mutations, okay, occur randomly from spontaneous errors during DNA replication or cell division. So sometimes when a cell divides, because we talked about cell division in the past lessons, it just makes an error and a mutation occurs. All right, so here is an example of a DNA strand, normal, and then this is a DNA strand for mutations. Here are the mutations. That is spontaneous. Induced mutations result from physical chemical damage to the DNA from outside factors. All right, so spontaneous happens on its own, randomly in the body, DNA gets replicated, and an error happens. Induced mutations are results of something external in forms of a damage to the DNA. So mutagens, or things called mutagenic agents that cause this mutation, are things like electromagnetic radiation. So X-rays, UV rays like sun, smoking can cause an induced mutation, or chemical exposure in large amounts to such as mercury. Okay, these can cause mutations in our DNA sequence. Since proteins are made according to the nucleotide sequence within a gene, so those nitrogenous base pairs, okay, mutations can alter the structure and function of the proteins. Like I said earlier, if you get a change in the genetic sequence, you're going to create proteins that are different than the normal, that are going to create a different function. So an example of a mutation is something called sickle cell anemia. It is a genetic disorder. It results from a mutation in the gene code, all right? So like a mutation in the gene code that creates a protein in specifically the red blood cells. So this is a genetic code mutation in red blood cells. Where specifically in the red blood cell? Something called the hemoglobin. All right. So the hemoglobin is important in the red blood cell because it helps carry oxygen. All right. This is a normal red blood cell. Kind of looks like a donut. All right. That's what its shape is supposed to be. A sickle cell anemia resulting from a genetic disorder because of mutation in the gene, the red blood cell doesn't really look like a donut. It kind of looks like a moon, all right? So the mutation goes from kind of like a donut to looking like this. And if the shape of the red blood cell changes, the hemoglobin can't carry the oxygen very well, all right? So this shape for the red blood cell, it doesn't carry oxygen very well. The oxygen can't bind to the iron, which is found on the hemoglobin, very well in that. So the oxygen decreases in its ability to be carried. The red blood cell, that's sickle cell, its ability, if it's in that shape, to carry oxygen decreases. And that's not very good because a lot of our cells require oxygen to carry out some of its functions. Not all mutations are bad. Some mutations can be good to the organism. They may help the organism. Sometimes they may help the organism to adapt to a change in the environment. All right? Maybe the organism can become faster, can be easier camouflaged, all right, from any kind of predator. Other mutations that occur in proteins of the DNA code, some codes don't make proteins. Those are called non-coding DNA genetic sequences. All right, so that's something that I just want to point out. Not all codes can make a protein. So if there's a mutation in the code for a gene that doesn't make a protein, well, then you're not going to create a protein that's going to carry out a function, and it's not going to affect the organism. It's called a neutral mutation. Okay? Some mutations can be harmful, such as the sickle cell anemia, which is kind of restricting the ability of the red blood cell to carry oxygen throughout the body. All right? You can watch this video on mutation if you want any further instruction. Okay, so not all genetic errors are a result of those point mutations, which I said was substitution, deletion, and I believe it was in substitution and deletion and insertion. All right? Other genetic sources of mutation occur on a larger scale, and those are chromosomal mutations. Chromosomal mutations are errors that involve the entire chromosome or part of a chromosome. So chromosomal mutations interfere, which can also contribute to the genetic disorder or infertility, ability to have children. There are two types of chromosomal errors. A change in the chromosome structure, okay, so maybe portion of the chromosome is lost or gained, or a change in the number of the chromosome. So an entire chromosome is either lost or gained. So an example of a chromosomal mutation is Down syndrome, or another way of calling Down syndrome is trisomy 21. So a person who has Down syndrome is born with 3 copies of chromosome 21. So do you remember when I showed you the karyotype up here? All right. Okay, this is a karyotype of a human. This is the karyotype down here of a person with Down syndrome. So if you notice, in this person who doesn't have Down syndrome, the 21st chromosome has 2. So this 21st pair chromosome has 2 chromosomes, and on the Down syndrome one, the person with Down syndrome, there are 3. All right, so that's why they call it trisomy 21 sometimes. So that extra chromosome on the 21st pair can cause different kind of genetic things. So that's where the Down syndrome comes from. As a result, we have 27 chromosome pairs rather than 46 chromosomes. All right, they have that extra chromosome. Okay, and rather than 26 chromosome pairs. Sorry, guys. All right. Okay, so here are some practice problems you can work on. So it goes down, you can fill those questions in. All right. This is a LearnLink gizmo. So you log in to your My Site, you go to your LMS grade 10, our page, and then you can select gizmos and you can launch your gizmo. I'll give you further instructions on this before, but this is the homework and worksheets that you can fill in and work on, and I will be giving you the solutions to that later on. Okay?